All right, I'll get started. The title of my talk today is going to be Cyclic Voltammetry of Biofilms. Up front, I wanted to just recognize that all of the work was done during my PhD and also as a postdoc at Washington State University, and it was funded by the Office of Naval Research. CV at Biofilms is a very broad topic, and so I've kind of focused down what we're going to talk about. And really, I've kind of written or put together this presentation considering a biofilm researcher interested in using electrochemical techniques, or maybe wanting to know a little bit more about cyclic voltammetry in general. We're not covering biofilms acting as a passive barrier, or we're not covering reactor or material selection or even scale up. I think those are specific topics. And of course, it's not just a literature review. What I'm trying to achieve here is just to give you a practical framework on how to utilize CV for biofilm research, and maybe highlight some of the difficulties you might face when you try to look at the classical CV description in textbooks and then try to apply that to biofilms. And so before we dive in, I wanted to just point out that most of this content is actually covered in Chapter 5 of this book on biofilms and bioelectrochemical systems. And also we have an annual course that's been, we spend about 4 days just talking about biofilms and electron transfer in both directions, and that might be of interest to you or to anybody in the audience. So with that, I'll start. We'll start simple. I just have a handful of basic introductory slides, and we'll get into CV. But what is a biofilm? For those of you who don't know, or maybe are new to this concept, a biofilm is basically microbes attached to a surface. And so that normally occurs with an initial attachment where the cells actually contact that surface, and followed by some kind of irreversible attachment, which means that the cells, once they touch down on a surface, can no longer leave that surface. That doesn't mean that they're dead. It means that now they're going to proliferate on that surface, and and that's usually called maturation. And then that maturation follows some kind of dispersion and kind of a cyclical process. And a simple internet search will give you lots of photos of what this kind of biofilm growth cycle looks like. The growth can occur on surfaces, or microbes or cells can attach to each other. The cells in a biofilm can be alive, dormant, right? Dormant means metabolically inactive or dead. And the cell debris and other biological matter make up what we call EPS. And the EPS is also known as the biofilm matrix. It's what surrounds the cells and is really a characteristic of biofilms in general. And I think people who study biofilms are really interested in EPS. And of course, there are void spaces inside biofilm, so that has to be taken into consideration. So if you construct this idea in your head that a biofilm is really a film of essentially cells, EPS, and some free void spaces, right? So why do we care about biofilms? I think for me, what's interesting about biofilms, and maybe to others, is that a lot of the complex interactions that occur in a biofilm and between cells lead to really unexpected outcomes. And a great example is what brings us here, right? Which is electrically conductive biofilms. I mean, who knew that you could find biofilms in nature that could actually conduct electrons across 100 microns? And I think now there are cable bacteria that do this over a centimeter, so millimeter. So it's very interesting. And biofilms, as we know, are often key players in energy, water, and the environment. And so we need to understand what that role is. And perhaps, that role, or what they do in terms of energy transformation, can inspire new biotechnology. And so these are kind of a few of the things as to why you might actually want to know more about biofilms and see how they can do things. So how do biofilms and electrochemistry get together? How do these fields kind of intersect? Well, electrochemistry, as a tool can be used to detect biofilms. It can be used to remove them. You can also stimulate biofilm growth and enrich for certain behaviors using electrochemistry, right? And so I won't introduce electrochemistry here. I just assume that most people have a working knowledge of it. But basically, you can use either a voltage or a current to induce some kind of change in a biofilm. And so that's interesting, right? Because then that ties into, some kind of the how biofilms handle electrons and electron flow, right? So in other words, if you wanted to study biofilms with electrochemistry and maybe use a technique like cyclic voltammetry, right, you have to understand how electron transfer works in biofilm. And maybe the overall goal in doing so is to modulate and control that biofilm. And that could also mean that you want to modulate and control the environment within the biofilm, right? So some of the critical questions that might come up, and I think about this a lot, or I used to anyhow, which was, if you considered biofilms as cells attached to a surface with the EPS over it, and they kind of form this protective layer that is different from the bulk environment, right? Where does the electron transfer happen in a biofilm? Is it at the top, at the bottom, inside, or just directly outside, right? And secondly, biofilms can be one cell thick, they can be hundreds of cells thick. So how do we understand or associate what happens at the molecular level, right? How does that impact the overall behavior that we see with the biofilm? And how is that related to electron transport, right? And third, what do we actually observe when we try to quantify electron transfer using an electrode? And that's this third question that I'd like to go over today, because the first two are actually quite difficult. So let's think about electron transfer in biofilms. So I have a very ignorant and naive concept in my mind that, if you had a cell on, let me get the pointer. If you had a cell in orange here attached to some electrode surface, which is this rectangle here, right? Somehow, let's say it's turning over acetate into some carbon and hydrogen and oxygen compounds, and along the way, an electron enters the cell. And this electron, I have no idea where this is actually occurring in a real cell, but somehow that electron has to get into the cell. And then at this point, you can consider it could take two paths, right? It could take a path through the cell somehow, maybe with some transporters, or it could go along the membrane, right? But ultimately, right, if you want the electrode to detect the cell, or if you want to understand electron transport through this biofilm, you'd ultimately want that electron to end up at the electrode surface and then eventually out. So not only does the electron have to go from its initial location through two separate paths, but it has to at some point cross over from the cell through the cell membrane onto the electrode surface. So that's why here there are two boxes, an initial electron transfer event into the cell from the electron donor, and then eventually to the electrode, right? And so conceptually, this is very ignorant, but it's interesting to think about this because, you might want to understand what this rate of electron transport is. Additionally, you might even consider the reverse scenario where, you would like an electron to come from the electrode, then somehow make its way into the cell, and then take these paths in reverse, and then ultimately take some kind of carbon, hydrogen, or oxygen compound and make something useful like acetate, right? And so electron transfer in biofilms, and maybe why you're here today, is you really want to understand how an electrode or how a biofilm or cells on a surface interact with an electrode, so that you may facilitate things such as acetate oxidation, or go in reverse and try to actually reduce CO2, right? And interestingly, I found a paper about this just casually, exactly talking about how certain anaerobic bacteria will reduce CO2 to acetate. So the question is, how do you get this to happen on an electrode? And I think that is really the focus of the field of electron transfer in biofilms and biofilm electrochemistry. I will say, because I have to speed up, but I did not really consider anything such as the EPS or, mass transfer resistance. But so, as biofilm researchers, you're really interested in thinking about these kinds of conceptualized pictures or schematics in your head. But in reality, right, when you actually try to go and do this in the lab, you have to compete with a couple of factors. And one of those factors, right, is the measurement system. So every time that you go and try to measure electron transfer in some way, or measure a voltage or measure a current, you're always up against the background. And this background is highly dependent on how you set up your cell. And so I'll briefly jump into that because I really want to get to CVs. But in terms of how you can set up a biofilm in an electrochemical cell, there are typically 4 ways to do so. The first one is on the top left. A is your traditional microbial fuel cell configuration. You pair a cathode, usually with a bioanode, microbial anode, right, separated by a membrane. Your cathode is typically some kind of abiotic reaction, such as oxygen reduction, right? And so you pair that to an anode where you have acetate oxidation. There's a voltage difference. You apply a load that's usually a resistor, and you measure that voltage. So what happens is, one, you're able to measure the cell voltage, current, and calculate power. And so this is, I think many people would be familiar with this. The drawback here is that you really don't know anything about the cathode and anode potentials. It's actually difficult to determine some electrochemical reasoning or mechanism for an increase in power. And in general, you can't study anything about the biofilm in this scenario, right? If you add a reference electrode, the picture changes. You can actually calculate the cathode and anode potentials over time. You can calculate overpotentials and potential losses, and you can determine a current limiting electrode. And in this case, this is your absolutely minimum setup to study electron transfer in biofilms. And the reason is really on this number line here, where you can actually calculate the cell potential under load and then understand what the loss is from the open circuit potential. But in order to run CV, you need a potentiostat, right? Because a potentiostat allows for absolute control of the electrode potential, or in this case here, if you look C and D, BE is a biofilm electrode, SE is a secondary electrode, right? And the potentiostat allows you to precisely control the biofilm electrode with respect to a reference, right? And I do hear oftentimes that people say a potentiostat can allow you to, you can damage your biofilm, or you can apply unrealistic voltages, but truthfully, that's not a good reason not to use a potentiostat, because as you'll see for the rest of this presentation, potentiostats, give you so much more information, and it's more advantageous to use one than to not use one because of this reason. So with that, let's get started on talking about one particular case study or one particular system, Geobacter sulfurreducens biofilm on electrodes. It is a popular microbial system to study. Right? Geobacter sulfurreducens is an oxygen-intolerant anaerobic bacteria that is able to grow on biofilms as electrodes. On electrodes as biofilm. And in general, they metabolize acetate, and the acetate in the presence of biofilms goes to carbon dioxide, protons, and electrons. It's a very simplified overall half reaction. the electrode itself acts as the terminal electron acceptor. And in this case, when you want to grow the biofilm on the electrode, there is no soluble electron acceptor. So there's no fumarate, for example. And so in general, the biofilm is only found on, in this case, a gold electrode. And it's more visible if you actually scrape some of it off. And so it's not anywhere else except on that electrode. So what happens is the current that you're measuring with the biofilm, right, it's actually a proxy for the biofilm respiration rate. And so that respiration is activated at voltages or applied potentials above about minus 0.4 volts versus silver chloride, right? The working electrode, essentially, if you construct that a biofilm reactor, it needs to be polarized above this to actually get the biofilm to grow. So what happens is that you grow, or you apply the voltage, in this case 0 volts versus a silver chloride reference electrode, and you add your inoculum. So you add planktonic Geobacter cells to, in this case, an EQCM cell. You can ignore that. It's basically just a standard electrochemical cell, and you monitor the current over time. So the key is, the electrode is continuously polarized to this value 0, and you introduce your cells at time 0, and over time you'll start to see that the current, right, which is a logarithmic, it's logarithmic here, increases over time, and eventually it goes from some kind of lag phase to an exponential growth phase here. And so x-axis is time here. So what happens is, as the current is increasing, that, and you can visually see this, or you can maybe take samples of the biofilm, or you can do multiple replicates. You'll know with multiple experiments that as the current is increasing, there are more cells attached to the surface. So the biofilm growth, again, is tied to the current. And current in general is a proxy for biofilm, the respiration rate, right? So during this entire process, you might consider, hey, my potentiostat has a CV enabled, and I can probably run it. And so, you stop the polarization, and the biofilm is just sitting there, and you can run the script, and you can choose maybe an initial scan rate of 30 millivolts per second. And you get this plot on the right, a voltammogram, right? So voltammogram is a current on the y-axis and voltage, or the electrode potential, on the x-axis. And what happens is you start at some initial value, voltage, and you start sweeping at a linear rate, and you start to see a sharp increase in current, and then you sweep back, and you notice that you end up at the same location. And you can do this at 2 different times, right? So you can do it at time 1 and time 2, where the current is at an increasing value, and you can see that you see 2 different types of voltammograms, one that is taller than the other. And so this is kind of like, okay, so what's next? So what does a biofilm CV tell you, right? And maybe at first glance, if you're looking at the voltage and the current, you can immediately say, well, the CV will definitely tell you something about the electrochemical behavior of that biofilm with respect to a known reference potential, right? And so how do you kind of think through what a CV is? Well, what happens is you apply a voltage, right? In this case, I told you that for this particular biofilm, a half reaction is activated above a certain threshold value. And so I know what value to apply. But in other cases, in other biofilm systems, you may not know what that applied potential should be. So if you go to apply a voltage, you may or may not actually cause current to flow. And because of this, right, you have to select a wide range of applied potentials to test. And your aim is to cause current to flow, to see if you can induce electron transfer within your biofilm, right? And then, with those ranges of applied potentials, you'll measure a resulting current, and you want to relate the two. And eventually, right, when you look at the current and potential relationship, that tells you something about what kind of mechanisms are controlling electron transfer in the biofilm. And in this case, oops, we can think about this a little bit more thoroughly. So let's say we chose a range of polarization potentials to determine a steady state current, right? So what that means is I arbitrarily decided on 10 different polarization potentials. And what I did was I set up 10 individual experiments, and I set the voltage, and then I added cells to each one, and I just watched this over time. And at the end of several replicate experiments, I decided to plot all of this. And I said, wow, this is interesting. So at certain voltages, I got to see current, steady state current. And at higher voltages or more positive voltages, I saw more and more positive current, right? And so you can actually get very similar amounts of information or a similar curve with just actual constant potential experiments, right? The problem is, right, it's really slow and it's tedious. And is there a faster way to do it? And that's what a CV, cyclic voltammetry gives you, right? So you can run this experiment within 5 minutes and understand that, if you went back into the literature and calculated a formal potential for acetate oxidation, you would get a value of about minus 483 millivolts versus silver chloride, right? Keep in mind that the formal potential is adjusted for pH and the temperature of the operating, how you grow your biofilm, right? And you look at where minus 483 is on your x-axis. It's about here, and I know this is 0 here, but right here, right where you would expect to see acetate oxidation, the current goes positive. And then at some point, right then, now you can say that, oh, this is interesting. So I only get current after the acetate oxidation half reaction is allowed, right, or activated. So that is kind of a general introduction into why you'd want to run cyclic voltammetry for biofilms, right? Is to learn a little bit about what's causing the electron transfer to occur. Is it acetate oxidation, for example? So the utility of CV is really, you're trying to support some kind of hypothesized electron transfer mechanism, and you do that by relying on some perceived changes in the voltammogram. You can vary temperature, strain type, biofilm type, salinity, the electrode material, the surface area. I mean, there's so many different parameters that you can change, and you have to rely on basically how does that CV change. But if you rely on how that CV or that voltammogram changes, it's really important that you implement CV correctly for any given system that you set up. And so that's where, I'm going to talk a little bit. The rest of my talk is going to cover this. Okay. So the textbook explanation for cyclic voltammetry is that it is essentially applying a triangular wave, in different cycles. So that's your panel A, where you start at some V initial at some slope, which is a dV dt, and you sweep from V initial to V reversal, at some time, reversal time. In this case, it's 50 seconds. And then you sweep back, and you do this multiple cycles until you hit a V final. And you're monitoring the current as you go. There's some initial charging current or current transient that occurs on the first scan, and your current goes through what looks like really complex behavior. The voltammogram comes when you plot the voltage and the current together, and this is what you get, the duck, or the normal redox behavior that you'd see in a textbook, which is an anodic peak coupled to a cathodic peak, and you can calculate all sorts of behavior. In this case, this is the current transient, and we're actually going in this direction. Okay? And you can calculate things from this. What can you calculate? Well, I'll get to it. But in a typical setup, right, you define these values. So if you've used the potentiostat in the past, if you're using one of ours, it's not This is a familiar screen. I won't spend too much time on it, but normally you define your scan limits, the scan rate, the number of data points per, the number of data points within the window, and the number of cycles you want to run, okay? And based off of that, typically for a reversible one-step redox couple from oxidized to reduced at 25C, you typically take a look at these diagnostic parameters. You can calculate the peak currents for both the anode and the cathode, the top two here, based off of a bunch of parameters. You can calculate that the peak differences are going to be about 58 millivolts for at least the one electron transfer reaction. The formal potential is going to be the average of the two, and so you can go on and kind of work with that. One of the difficulties, though, with applying this, these simple analyses is that biofilms are a heterogeneous structure. They restrict diffusion. And if you're curious about diffusion in biofilms, I would recommend taking a look at this paper here. We actually measured relative diffusion coefficients within certain biofilms respiring on electrodes. So the D here of the oxidized mediator, in this case, right, it's really poorly defined in biofilm. What is that? You'd have to measure it. Other things that are difficult to understand or measure is even simple things such as pH. pH inside the biofilm is significantly different than pH outside of the biofilm. And what's relevant to electron transport is unfortunately the pH within the biofilm. But you have no way of knowing that unless you measure it directly, right? So that's a challenge. Other things is that we actually don't know what the concentration of the redox compounds are in a biofilm because the biofilm produces it themselves, and two, we don't know where they are. So when you try to apply, a simple textbook equations to the biofilm scenario or the biofilm case, it's really difficult. You don't know what the bulk concentrations or what the biofilm concentrations should be. So what can you do? How do you know if your CV data is right or wrong? And that's where, spending time to really understand your system and your cell, even before you start to work with the biofilm, it's important. First, you want to understand what the baseline working electrode response is. Second, you have to understand what your potentiostat is doing. I won't really cover the second one today. I would really highly encourage you to contact us if you do have questions about what the potentiostat is doing, and we'll tell you about it. Third, you want to test your setup with a known redox system. In this case here, I actually chose ferrocyanide in order to explain what's happening in my electrochemical cell, and you should do the same. It has nothing to do with biofilm, but you'll be better off understanding what you're measuring if you know that if you know how to measure what's going on in a known redox system. Fourth, you want to test your setup in the absence or with an abiotic control, which is basically the absence of cells. And fifth, if you can find one, is test your setup with a biofilm proxy. So let's start with the first bullet. How do you understand the baseline working electrode response? Well, you define two ideal working electrode behaviors. This comes out of Barton Faulkner, but there is a polarizable electrode and a non-polarizable electrode. In this case, it refers to if you make a voltammogram or run a CV on your working electrode in a standard electrolyte, you can consider saline as one, or PBS, phosphate buffered saline. And if you run it and you see it's mostly horizontal, it's polarizable. If it's mostly vertical, it's non-polarizable, right? And these are the two idealized types, right? Your ideal working electrode, it must be polarizable, right? And the reason is because you want zero current to flow in the absence of what you're trying to study. And so in this case, what you're trying to study is the biofilm. So you want a polarizable electrode in the absence of biofilm. And so the polarizable region is also called the working region for a working electrode. Unfortunately, there is no predetermined theoretical calculation for the working region. So how do you find it? I would read this paper. I'm slightly behind, but essentially you start wide and you start decreasing that potential window until you see zero current or baseline current. And the factors that affect, this kind of analysis and how you it is listed here. I will say that, and again, from the same paper, if you look at different materials, you'll see different windows. And a lot of this is bounded by basically water electrolysis, but you can see how that shifts in sulfuric acid, for example. So the key is find your working region of your electrode. So when I was doing this work way back when, here's an example of glassy carbon in, I think, PBS. I ran this in the presence of oxygen. This is the oxygen signal here. And realistically, I could only measure some current from a biofilm in this region, right? So the next step is to understand that when current flows to your electrode, it follows two paths. And this green, again, is a part of what I discussed earlier about this background interfering with you trying to measure what's important to you, which is the cell or the biofilm, right? When current flows, it goes through either a Faraday path or a non-Faraday capacitive path. And in both instances, that current has to flow through some kind of uncompensated resistance. So what are the implications of, first of all, the capacitive current? Well, it tells us, right, that whenever we run a CV, we should expect that there is an initial charging current, right? So you should always see this kind of tail coming off of a CV if you don't condition your electrode. So that is the consequence of charging. It also means that when you run a CV, there is a hysteresis. And that hysteresis depends on, hysteresis means from here to here, basically these red lines. But that hysteresis is dependent on the scan rate. And that can be a very big or tough issue if you have very high surface area electrodes, which is common in biofilm studies. Most people tend to use some kind of carbon cloth, carbon felt, or, and they tend to be huge. Well, what happens is at certain scan rates, your non-Faraday current is greater than your Faraday or your biofilm current. So in other words, your background suppresses what you're trying to measure and your experiment fails, right? What happens in terms of the uncompensated resistance? Well, if you had to draw an equivalent electrical circuit analogy of the cell where you're going to grow your biofilm, and the details are here on our app note about IR compensation, you'd realize that there's what your potential that measures is here, and the current you measure is also here. And what you really want to know is actually the points between E and F. And that's what is, electrochemically relevant or relevant to your biofilm. And how does that impact you? Well, if you know that your potentiostat is measuring this value across an electrometer, and that is different from the potential of the electrochemical interface that you want to know about, in this case, your biofilm, you have to offset what the potentiostat is measuring by what's called the IR voltage drop, okay? And this can be a challenge if you scale up, or if you're in very dilute, if you have very little electrolyte in your system. So one very good example is when you want to go for some kind of post biofilm analyses where you have to put your sample in water or DI water or something. You can't make good voltage measurements. You can't run CV in these types of systems because of this IR. And I would encourage you to take a look at what are the consequences and how you can kind of overcome the uncompensated resistance. So, I mean, to sum up to this point, before starting any biofilm experiment, I would highly recommend that you measure and understand the working region of your electrode, measure and record RU, the uncompensated resistance, and run several background CVs to plot along with your future biofilm CVs and several scan rates. And that leads us into really what is the initial CV parameters. And I'm just going to pause for a second. This is just try this initially and iterate on this. But since there's going to be a recording, I'm going to pass through that. Breaking down a biofilm CV, I mean, you can take a look at, so this is a Geobacter biofilm CV, right? There are a couple of different regions of interest, and I've also plotted the counter electrode potential that was measured during the CV. You first start in a region where there is no Faradaic reaction, you don't see any current, and it's basically zero versus background. Then you start to see an initial current response. Usually that it contains the open circuit potential. And as you scan, you activate the biofilm response and see this region usually contains the half wave E1/2 potential, where the slope is at a maximum and the charge transfer resistance is at a minimum. And then at D, you'll start to approach the limiting current and eventually get there. So normally when you frame a CV, you want to capture a region of no current to the limiting current. And at the same time, what's happening at your counter electrode is you're evolving hydrogen, right? And so this can be problematic because biofilms and anaerobic bacteria such as Geobacter can actually utilize hydrogen as an electron donor, or an electron source. So this can result in some kind of parasitic redox cycling in your cell, and that should be avoided. But we don't really have time to go over that. So CVs can be kind of binned into at least two different types. There's the turnover CV, which refers to microbial turnover of the electron donor, acetate. And what happens is when you have more and more biofilm, right, the characteristic sigmoidal shape kind of becomes more visible, I would say. And what happens is the catalytic current, which is the current that's caused by the renewal of the redox mediator in the biofilm, right, you start to see catalytic current rise over time. The general shape of this turnover CV, right, the sigmoidal shape, is retained, but the catalytic current increases as more and more biofilm. So this is the standard plot. You'll see this a lot in the literature. The other type of CV is a non-turnover CV, where if you had a mature biofilm, you could essentially dilute out or wash out acetate or the electron donor and watch how, as the catalytic current is basically suppressed because there is no longer any microbial turnover. And what happens is the sigmoid shape is lost, and you start to get, the traditional voltammogram with various redox peaks. So here you can see the transition on this voltammogram, and here is where finally, after a day or so of absence of under starving conditions, you'll start to see redox peaks. Right? All right. So the types of CV analysis that you can see that you get are typically mostly it's scan rate analysis. You plot the peak current versus scan rate, right? And you can do this both conditions. And then there's also one that's less used. It's a peak coupling, and I'll go over that in a little bit. Initially, if we look at a scan rate analysis for turnover CVs, you'll see that one, you see the consequence or the hysteresis increase with scan rate, right? So you know that there is a capacitive behavior here for this biofilm. I'm increasing the scan rate from 1 millivolt to 100 millivolts per second. And typically, you measure these peaks and you plot them for the anodic peak and the cathodic peak, and you watch how that varies with the square root of the scan rate. I'll cover why that is in a second. And you can do this also under non-turnover conditions, where here I'm going from 1 millivolt per second to 320. And you can see again, these redox peaks are increasing with time. So, right, one of the challenges with this type of analysis, right, and it's different from what you'd see in the textbook, right, it's almost impossible to actually resolve the individual peak behavior, because as you can see in this voltammogram here, where I've normalized the current to the peak current here, you can see that when I go from 1 millivolt per second to 10 millivolt per second, you can see that 1 or 2 peaks, or maybe 3, turn into 1 overall peak. And it's even worse on the cathodic sweep, where 1 peak and this really oddly shaped peak here turn into 1 large peak. Right? So going from 1 to 320, you know, I don't know what how valid actually this Gamry analysis is, and you have to make assumptions about how these individual peaks superimpose onto each other, right? And what's the theory behind this? I mean, can you use, you know, these relations here where it's a bunch of constants with scan rate to the 1/2? I mean, how valid is this when you look at biofilms like peaks that are tightly packed, such as this? If you actually extend this, so here is a non-turnover CVs for the Geobacter biofilm, the same ones, but I'm showing it from 0 or 1 millivolt per second all the way up to 1.2 volts per second. Right here, you can see that the peak currents plotted versus the square root of the scan rate kind of looks linear. Right, so it kind of seems to show what we'd expect from this relationship here, but at the same time, it's really not linear. And whereas if you plot it versus just the scan rate, it's very nonlinear. And if you look at the peak potentials, I mean, essentially the takeaway from this graph is that, you know, the actual behavior that you'll see in Gamry analysis with biofilms is going to be vastly different from what you'd see in a textbook. And you have to be very careful about applying what you see for standard CV analysis to what you see in the biofilm. And a lot of people in the literature make this mistake of seeing a straight line and then assuming that it's a diffusion, or, you know, making basically claims about this. Peak coupling is interesting. You can take the same non-turnover CV and you can choose smaller scan windows, and you can see how peaks are coupled. So on this blue trace, you can see that if you start here, right, you can actually run through this last anodic peak and get back the complete cathodic peak here. Whereas if you scanned here in the middle, in this black one, you actually suppress these last 2 anodic cathodic pairs and are looking at what really happens here. If you have questions about this, I don't have too much time for it, but please contact me directly, and I can talk to you individually about whether or not this could be useful for your setup. You know, where CV gets really interesting, and I know we definitely don't have time to go over the details here, but it gets very interesting when you start to couple it with other techniques. And a good example is when, for example, which was my previous webinar about biofilms and QCM, is that you can run a CV in young and mature biofilm. In this case, it's still Geobacter biofilms. You can compare it to some baseline response, and you can see a frequency shift. And a frequency shift, if you don't know, for QCM means kind of a mass change, just for, you know, for our considerations in this talk. So this is telling me right now that, you know, as you run a CV, there is some kind of apparent mass change in this biofilm. And I think that's quite amazing, actually, because if a CV can be run in 5 minutes, it's almost like saying the biofilm increased in mass within that time frame, which is, you know, quite interesting. You can couple a CV to an RDE. So here, you know, your turnover CV. So this is the respiration rate. You know, you can rotate and you can cause the current to increase. And at these different rotation rates on the right here, you can superimpose your turnover CVs, and you can see that, you know, under no rotation, you got some kind of diffusion limited current. And with rotation, you've got some kind of rotation limited current. And can't go into the details now, but essentially what is interesting here is that you retain the sigmoid shape, but the catalytic current increases. And if you know anything about Geobacter biofilms, you'll know that, you know, mass transport is supposedly not a limiting factor with these biofilms, but for some reason we can actually measure an increase in the current. So that's actually another interesting kind of coupling for CV and rotating disc electrodes. And finally, I'll just say that I've been talking about Geobacter biofilms, anodic biofilms. What does it look like for other types of biofilms? So let's say you had a cathodic biofilm. And in a cathodic biofilm, you actually measure cathodic negative current, like on panel A here, where you sweep from positive potential and towards a negative. And in this case, here's my baseline response without the biofilm. And with the biofilm, I can start to see cathodic current show up here. At the same time, I'm measuring oxygen close to the electrode surface. And here you can see that this cathodic current is tied to oxygen reduction. And this is also tied to oxygen reduction. Whereas here in red, right, this is tied to the cathodic electron transfer mechanism of the biofilm. And so, you know, whether you study anodic or cathodic biofilms, right, a lot of the analysis that I've been talking about, like this is a sigmoid in reverse, right, it all applies. So with that, luckily I ended in time. I'll say that, you know, when implemented properly, you know, CV and cyclic voltammetry is definitely a powerful tool in guiding how we develop hypotheses for electron transport in biofilms. And although I really barely scratched the surface, one of the things that you can look forward to after learning about CV is actually coupling it to different techniques. And there, by coupling it to different techniques, you can gain access to different, I would say, aspects of the electron transfer model within biofilms. And that's it. Questions?
Okay, thank you, Jerome. That's some really interesting electrochemistry there. Poses a lot of questions. So, okay, everybody, we'd be happy to take any questions that you might have. Just raise your hand and we can unmute you, or you can type it into chat, and you know, we can see if we can get Jerome to help you out. Okay, Catherine, you should be unmuted if you would like to ask your question.
Sure. Thanks, Jerome, for that talk. It was a good complement to that book chapter. I have a question for, you know, breaking with the biofilm. You have those multi-peaks sort of all crammed into one, you know, area of activation, basically. I have a question. Do you have any suggestions for what you can do if you realize that the species that you're looking at isn't really reversible? I mean, you talked about how complicated the biofilms could be. You know, what if I only ever see the cathodic peak and I never see the oxidation peak? I don't know. Do you have any tips for analysis of that sort of thing?
You might, have you tried just varying the scan rate like dramatically?
Yeah.
Yeah, and that didn't show the anodic peak at all?
It's sort of like sometimes it feels like it, sometimes it doesn't. Could obviously be coupling to the electrode and things like that. But do you think doing the short scans, instead of doing the full range, just kind of focusing in on the short scans at a really slow scan rate, you think that might
would help, right?
Tease out. Yeah.
So it would help. I would have to look at what you've measured to see how to formulate kind of like a plan of attack. If you just see the cathodic peaks, it's interesting because it seems like whatever you're measuring is, you know, that anodic process might not be coupled to the electrode at all. You might never see it.
Yeah.
Right? And that's interesting. That means some things for what's going on within the system you're studying. So, I mean, that's a very specific question. we could talk about it afterwards if you'd like.
Thanks.
But yes, if you only see like a cathodic peak or you only see an anodic peak, it's not necessarily it doesn't mean that that anodic process doesn't exist. It just means it's separated from your electrode. And then that kind of leads back to that question about where does that electron transfer happen, right? So.
Okay, Jerome, we have a question now. Could these measurements be related to biofilm corrosion at all? Could you use these types of measurements to study biofilm induced corrosion?
So yes and no. Yes, because I think if you were studying biofilm corrosion or maybe like microbially influenced corrosion, you'd want to kind of understand how the biofilm responds, maybe in the absence of the corrosion process. But in an actively corroding system, applying the wide voltages that you see in here might change the corrosion mechanism. You know, that's my main worry there. And that's it's difficult to say. So essentially, if the corrosion happens during the biofilm respiration, I guess, it would be okay. Yeah, I don't know. That's a tough question, Chris.
You essentially need to starve the biofilm to prevent the corrosion. Is that what
right?
you're
Well, the thing is, right, the biofilm's alive. So if you starve it and it engages in some kind of, I don't know, upregulates some other metabolic process, then that changes the corrosion, right? So it's not easy. So it's hard to say. I think like most biofilm problems, you have to attack each case specifically because each one has a unique metabolism and molecular machinery that it uses. So.
Okay. Now the next question that I see here is it possible to measure CV of the biofilm after the electrodes are decoupled from the MFC?
Yes. I could think of it physically, like you could actually remove it and then run it in some other vessel. Or you could just remove the load and remove all electronics, then hook it up to a potentiostat and run the CV. You know, the biofilm, it takes a long time for it to accumulate on the electrode, assuming you've run your MFC for a while. And in this case, removing the load to hook up a potentiostat and run the CV shouldn't really harm your biofilm too much, unless of course you ran a very wide CV and you electrolyzed some water and you generated a bunch of bubbles and you basically delaminate your biofilm. I've done that. You don't want to do that. But so the answer is yes.
Okay. And we have another one here. What if the strain is not really electroactive? What can one do to optimize this?
So if the strain is not electroactive, in other words, it as is or in a native state, or you somehow manage to force it to grow biofilm on an electrode, and you start to run a CV and you see no peaks, no ferritic response, just background, you can try to add mediators that are not toxic to the biofilm, and you want to choose mediators that have varying standard reduction potentials and see if you can't get the biofilm to indirectly reduce those mediators that can then be detected on the electrode. And so you can use mediators as a bridge. And I think this has been done for some biofilms, some E. coli biofilms, others as well. I think this is a big problem in electrofermentation, the CO2 reduction type deal, but I would try that. If that doesn't work, then you can consider having a mediator that doesn't react with the biofilm, but actually reacts only with the electrode. And as the biofilm grows, it will block access, and you can see a reduction in the peak currents as the biofilm grows. And this is kind of a reverse kind of electron transport deal where a decrease in peak current will tell you more about the biofilm. It's possible, I don't know. These are just different ways to attack that problem. But,
okay. Now we have another question. Are there any other kind of non-electrochemical measurements to see how continuous these films are?
In a set, are we talking about like structure? I mean, I know you don't have the question, but
I don't. But if Richard, if you could provide a little bit of follow-up, we might be able to get an answer there. So we'll wait to see if he responds.
So I will say though that, I mean, you would tend to use some kind of confocal microscopy or some kind of light-based technique to couple maybe the thickness of the film or the density of the film with respect to the current that it's producing or the peaks that it's exhibiting on the CV. In that sense, you'd need to have some kind of strain that is observable by a microscope, right? Or have a setup that allows you to do so, kind of like a flow cell setup. So at this point, it's kind of like a coupled technique again.
Okay.
Or you could use QCM.
Okay. Let's see, we have now another one. You mentioned running a control experiment in the absence of cells, but also with a proxy biofilm. Do you have any suggestions for a material for that, for this proxy biofilm?
I mean, you could try to use agar very simply, or some kind of hydrogel. So if you think of a biofilm as a hydrogel, and you can add different ingredients that a hydrogel to mimic certain features of your biofilm. I think in previous work that we did at Washington State University, I used some conductive polymers as a proxy for the conductive Geobacter biofilms that we were studying, especially on QCM. That's also a possibility. So basically any kind of film that you can get on the surface that is mostly water with lots of void spaces that restricts, kind of creates a kind of a microenvironment on top of the electrode would work, I think. Especially if you fixed certain mediators or compounds, organics within. So.
Okay. Let's see, we have a straightforward one. Can CV of biofilms be run on a 2-electrode system?
No. Because you need the reference electrode. I think if you ran it in a 2-electrode mode, as more current flows, the voltages and the voltage window moves in an unpredictable and unknown manner. And so the CV becomes useless. The important aspect of the entire analysis is that reference electrode and being able to link back to the half reactions that are tabulated in a lot of the standardized, I don't know, the CRC.
Okay.
Yeah.
Okay. Is there a simulation tool we can use to study the biofilms and its electrochemical characteristics?
I think different labs have attempted to do this. I don't know if anybody has really put out a software that could do this. I think the DigiElk that we have, Chris, could do this, but you'd have to specify a lot of different reaction steps. And then at that point, it becomes so complex that I don't know if it's a useful modeling software. You could take a look at some of the literature that describes or tries to fit a lot of the CV data empirically and use those equations, like the Monod I can't, the name's escaping me, but there are a few. If that person can reach out to me, I can talk about it after the fact. Probably reach out that way.
Okay. Yeah, I can imagine it's quite difficult to try to simulate these systems as complex as they are.
Yeah, there's a group in the Netherlands. I don't know where, but they've done like kind of cell-based, like individual cell modeling, but they, that's a lot of effort.
Yeah.
So.
Yeah. Okay. I have a question here. I'm not sure I fully understood. How difficult is it to prepare electrolyte for biofilm growth?
I think the electrolyte it has to be, it shouldn't be toxic, right, to the biofilm, and it shouldn't participate directly in the electron transfer that you're trying to study. But otherwise, you could just look up various growth media that is used in microbiology and kind of adapt it to remove certain metals and other easily oxidized and reduced compounds, and then use that. So, I mean, there are things like if you're studying biofilms and wastewater treatment, for example, there's artificial wastewater formulations. If you're in the ocean, there's artificial seawater. I mean, there's a lot of standardized salt solutions for biofilm growth or microbial growth that could act as an electrolyte for biofilm. Interestingly, if you wanted to kind of change the sizes of the ions, the cations and anions, I think doing some substitutions would be like an interesting route, but it would be very specific.
Yeah, okay. Okay, I think those are, unless there's any more questions, we have a couple that are pretty specific. So, oh wait, we're getting one last one here. But as I was saying, that the very specific questions will follow up individually. So make sure that you please submit your email address also. Jerome, we have one here on, can you go to slide 30, please?
Yes.
And here the question is, what did you mean by the working region of your electrode?
So in this case, the working region, as I described here, is essentially the region where you get no current response as you sweep the voltage. It's bounded a lot of times by water electrolysis, so hydrogen evolution on the low end and oxygen evolution on the high. In this case, right, you can see it for different, it's shifting, but essentially it's the flat region on your CV, right? And so in this case, when I just randomly ran CV on a glass carbon electrode and PDS, I didn't de-aerate the solution, so I had what's essentially is a flat region here that was basically tightly bound or tightly around zero. And then there was this region here, which is basically oxygen reduction. And so if you had a biofilm response here, redox, it would be superimposed and very difficult to study, right? So you wouldn't want to operate in this region, in this voltage region, which is minus 0.6 volts to minus 0.2. If you got rid of the oxygen, this would also be flat, but it's just an example.
Okay. I have another question. Can you quickly summarize what kind of data you can reliably get from performing CVs versus what kind of data you cannot trust? Such as midpoint potentials tend to be accurate, but it's hard to tell whether the system is diffusion limited.
Right. I think with just CVs alone, making claims about whether a system is diffusion limited is very difficult. What you can reliably tell from a CV is midpoint potentials, limiting current, and in particular, and I really unfortunately didn't have time to go over it, but this concept of the half wave potential right here, right? That's where the slope is at a maximum. That means that the charge transfer rate of your biofilm is at a minimum. Right? That says a lot. A couple of papers have described this as, you know, this is kind of a hallmark or a feature of conductive redox hopping so kind of like an underlying mechanism, right? So this E1/2 is actually important to track with different biofilms over the growth. I don't know, it's difficult to tease out here, but in actuality, during biofilm growth, the E1/2 doesn't change much. It's really surprising, but it's consistently about minus 340 millivolts versus a silver chloride. So it can actually be a good parameter to track. We just didn't have time to kind of dig into that.
Okay. Okay, let me think here. I don't know if it's related to these slides or not, but somebody would like to know if voltammetry can be used to determine the contributions of extracellular electron transfers used by the biofilm.
I think I would need more information or details about what they're referring to.
Okay, I have their email address. We can follow up separately.
Yeah.
Okay, we still have a couple. Are we still good on time? There's 3 questions here. Can you give an example of an electrode that is a non-polarizable electrode? And the second question is, what is the best scan rate for biofilm CV? What is a typical scan rate for biofilm CV? And should we stir the media when doing CV or no?
All right. The first one, I mean, it's relative, right? Because in looking at a non-polarizable electrode, right? It's basically anywhere where what's supposed to be a inert background electrolyte is no longer inert, right? So in this case, water, right? A good example is, you know, even a glassy carbon electrode can be considered non-polarizable in the presence of water if the voltage that you have to apply is between 1 volt and 2 volts out here, right? So that's considered non-polarizable, whereas in a different region it is polarizable. So it is relative, and so it doesn't really mean that glassy carbon or gold or platinum is always a polarizable or always a non-polarizable electrode. That's why you have to determine it with every new system and new setup that you have. I will say the second question, Chris, was the typical scan rates.
Yes.
It was here. Basically, start with 10 millivolts a second and go slow if you need. Slow can be 1 millivolt per second. Usually if you go at 100 millivolts per second, which is the default for most potentiostats, that's usually too fast. But so that would be typical. At least that's what I've used in the past. And the third question was?
Stirring the media.
That's a good question, and one that I didn't touch on. I mean, most of the textbook, the treatments of CV require that the solution be stagnant and quiescent. In my opinion, you know, biofilms are very sensitive to the mass transport and any mass transport in the solution. So if you do have stirring, and that's the way that you've grown your biofilm, I would recommend that you run your CV with that stirring on. As a backup, you could actually turn that stirring off and get that CV, but more often than not, you'll see that that is different, right? Because in the case of, for example, and that's what I was talking about in that very last slide with the CV. So RDE here, the rotation can mimic stirring. Right? It's a fancy way of stirring the biofilm. Mm-hmm. In this case, you can see that, you know, with and without that rotation, with and without stirring, you will see a change in the current. So in the end, this is one of those discrepancies that you would encounter between, you know, what you see in textbooks and what you need to do practically for biofilm work. Is this, make that decision and then account for it.
Okay. And let me see, we have, I think we have one more question at the moment. And I don't know if this is too much to get into here, but you mentioned the hydrogen evolution reaction as a problem.
Right.
Do you want to explain any more about that here, or is that kind of specific? And
no, so it's a very big problem.
Yeah.
It's just not tied to CV so much, right? So the problem is if you have anaerobic media, basically no oxygen, and there's no electron acceptors, and you're producing anodic current with your biofilm on the counter electrode that's in the same solution, or you know, just spatially separated, you're generating hydrogen. So that hydrogen is like super saturated above the counter electrode. It slowly makes its way over to your biofilm, and if that biofilm, like Geobacter, can actually use the hydrogen to generate more anodic current, then you can safely say, I mean, almost 100% guarantee, I've seen it in lab, that the current that you're measuring is also caused by hydrogen turnover. You know, and I don't think the biofilm gets any kind of energy. It doesn't grow just on hydrogen, but you will measure a current, anodic current, right? And that's going to interfere in your measurement. So the solution to it, and which I just glossed over at the very, very beginning, is to use a membrane, a semi-permeable membrane, to separate the hydrogen that's evolved in your counter or secondary electrode from what's going on in the biofilm. And I can send over my old paper about this. Yeah.
Okay. Then it looks like we have one more question here. Even though you talked about CV this entire time, is chronoamperometry a valid way to study biofilm electroactivity?
Yes, absolutely. It just wasn't the topic of my talk.
Yeah, okay.
Yeah, I mean, I just specifically CV, and that was kind of the reasoning why I mentioned this thought experiment. Actually, on some level, chronoamperometry and studying the steady state current of a biofilm is equally important, because, you know, a CV is a snapshot of the kinetics. But sometimes when you're looking at long-term installations or trying to understand how your biofilm behaves over days and months and years, actually a chronoamperometry or a constant voltage test is more important than a CV. CV is more important if you're trying to understand mechanisms, whereas chronoamperometry and long-term experiments help you understand kind of the evolution of your biofilm over time. And that can be interesting to certain people. So both equally important. I just didn't talk about it.
Okay. Sounds good. And I think with that, Andrew or David, did you guys have any questions?
Nope. No, none to add.
Okay. So I think with that, we're going to wrap it up, everybody. You know, thank you, Jerome, for this talk, and thank you, everybody, for attending today. We're going to have another one in a few weeks. We're going to go back and visit EIS and go over ways to deal with difficult samples, some very high impedance samples or high impedance reference electrodes or low impedance samples, and ways you can make better measurements there. So with that, thanks again. We're going to start putting people back into the waiting room so that we can take the time to save the chat and sort of wrap everything up here. The recording will be up on our website and YouTube channel, and we'll send a link to that tomorrow with our regular Friday focus. Thank you, everybody.